Okay, I try to look in the camera. Welcome everyone to our ICG Lab Talk series. It's our first talk in the semester. It's a pleasure to introduce Professor Stefan Bruckner from University of Bergen in Norway. Stefan did his computer science studies and also his PhD at the Technical University of Vienna. He finished in 2008, and then he was employed as an assistant professor at the Institute of Computer Graphics and Algorithms, also at Vienna University of Technology. His research is mostly focused on interactive visualization technique for biomedical data, illustrative visualization, volume rendering. It goes into this referred to as scientific visualization direction. Stefan was also awarded with the Eurographics Young Researchers Award in 2011, and yeah, he moved then a couple of years ago to University of Bergen to become full professor there. And now we're actually curious to hear more about the topic. So the title of the talk is Smart Visual Interfaces in Data Exploration and Analysis. The floor is yours, Stefan. Thank you.
Thank you very much, and thank you for the invitation. Hello to you people in Linz and the world, whoever may be watching. And let me switch over to my slides. Let's see whether this works. From my title side, I hope you can see that now. So what I want to talk about today is sort of sounds a bit arrogant. So smart visual interfaces in data exploration and analysis. So before I actually start, I have to add the obligatory quotes to my title, if it actually works. Yes, there they are. So these are the quotes, and I will, in the very beginning, qualify what I mean by smart. So smart, on the one hand, could mean visualization done by very smart people, and I can't claim the kind of smartness of some of those people that could be doing visualization. So I have to use another definition of smartness. And what I actually mean is, all joking aside, it's what is also known as knowledge-assisted or knowledge-based visualization. Essentially, the principle is that we want to incorporate domain knowledge into the design of our algorithms in order to make them do what the user expects them to do. So this knowledge can be manifest itself in different ways. So we can think about knowledge about the data that we may have. We may have certain information about data semantics, we may have certain information about features in the data, but it can also refer to knowledge about what the user wants to do in more general terms, and it can even be knowledge, sort of meta-knowledge about the algorithm that we are designing. So basically, we can also incorporate information about the algorithm's behavior into the algorithm itself. And the smartness simply comes from the effective use of this existing information. And to maybe split it up into a bit more detail, we can think about different levels of smartness in this context. And of course, these levels are not necessarily discrete, but it's much more of a continuum. Still, there are a couple of stages in this continuum that are probably useful to think about this in more concrete terms. So on the one hand, that there is the technique level. In most cases, when we are dealing with data in visualization, the data was generated for some reason. There was a specific purpose that, for example, CT or MRI scan was acquired, and we have typically knowledge about this reason, and hence this principal knowledge can already form part of the smartness that we can integrate into our algorithm. A second level, which is above that, if we think about this as a bit of a hierarchy, is the parameter level. So as computer scientists, we typically, if we don't know something, we add a free parameter and think this is done, and usually this is the only choice that we have. But then we end up necessarily with algorithms that have quite a lot of parameters. Now, but we have to recognize that not every part of the parameter space is equally useful, and also that these parameters may not linearly correspond to some of the things that the user may want to do. So we can also think about constraining the parameter space in a meaningful way. And the third sort of the top level of, at least in the sense that I want to talk about this today, is the interface level, where we actually exploit knowledge about the performance, not in efficiency, but rather in an effectiveness sense of our techniques or parameters. Now, after this rather abstract introduction, let me maybe set a scenario that is a bit more concrete and that I'll be focusing on today, simply because it's an area that quite a few people in scientific visualization have been working on, but I do claim a sort of a more general context for some of the things, for some of the principles at least, that I want to present today. So visualization in radiology is characterized by the fact that we are usually talking about the visualization of imaging, mostly 3D imaging data, such as CT, MRI, but also things like ultrasound, PET, and so on. And this data is typically stored in something that is called PACS, a picture archiving and communication system that is installed throughout the hospital. And all these different imaging devices, archive nodes, and also workstations that are used for visualization are interconnected using the DICOM standard, which is just the common communication protocol that is used there. Even more specifically, one area that is of interest is the investigation of blood vessels. In particular, in the leg area, where there are certain pathologies that can lead to death in the worst case, but also to the need for amputation of a limb, for instance. And these pathologies are often caused by the depositing of calcifications in blood vessels that build up and eventually build up to what is called a stenosis that reduces the amount of blood that can flow through the vessel. And eventually it may even completely stop the flow of blood in the interior of the vessel that is called the lumen, which is of course very problematic. And to detect these kinds of pathologies early is one important aspect of visualization of radiological data. And just to give you an example here, so typically CT scan, in this case a CT angiography scan that uses a contrast agent, has around 1,500 slices if acquired for this purpose. And the very basic way to investigate this data is to scroll through all these slices. So that's what the radiologist does, clicks forward and looks at all these slices. And as you can imagine, this is quite time consuming, but also requires a lot of concentration. And if you think about it, that the vessel may also or may also be located within the slice plane, then it can be quite difficult to mentally reconstruct the shape of this vessel and to assess whether there is still a viable lumen in this blood vessel. An additional complication is also that this is really a high throughput scenario. The radiologist typically in a clinical context has no time to play around and tune parameters, but he really has to diagnose one patient after the other. And hence, there is a real need to make this assessment better, and with better, I specifically mean the examination should be as safe but also as fast as possible without compromising safety, of course. So, question is now, of course, what can we do in order to avoid that we need to traverse all these slices? Well, one of the ideas is we are dealing with 3D data. Well, why don't we visualize this in 3D? Then we can avoid scrolling through all the slices. And of course, this has been done, and this has been investigated. And there are two standard methods for visualizing such data in 3D. One is called DVR, direct volume rendering, and there is another technique that is actually, I was just talking about that we can, of course, do a 3D visualization, and one of the common methods that has been used for this purpose is direct volume rendering, and the other method that is quite frequently used in practice is what is called maximum intensity projection. And here are just the images that these methods produce, and you see you get a very colorful, nice images for people who like computer graphics from DVR, and rather boring for people who like nice images, images from MIP. But it turns out that the situation is a bit more complex. So DVR, in essence, is a physically based method. It's essentially a simplified simulation of emission and absorption in participating media, while MIP is practically motivated, and it's simply based on the fact that when these examinations are made, they are done using the CT scans are done using a contrast agent. So there's a radio opaque substance injected into the patient, and this substance essentially causes the blood vessels to be very contrasted in the resulting CTA scan. And hence, it does make a lot of sense, since blood vessels will have higher data values in the reconstructed volume, to project the maximum value along each viewing ray. The other hand, DVR, on the other hand, will require quite complex transfer function, because we need to somehow specify the emission and absorption properties for data values in the volume, while MIP typically just suffices with a simple contrast adjustment. But what we get from DVR, as you saw, are shape cues and occlusion cues due to accumulation and shading, while MIP, since the maximum operator is order independent, causes or leads to spatial ambiguities. So, in essence, what we would like to have an improved visualization is something that combines the advantages and removes the disadvantages of both methods. So, can we incorporate MIP-like domain knowledge into the DVR algorithm? Well, to do that, we can take a look at how these algorithms actually behave. So, just to set the scenario, the way that these images are typically computed is to use volume raycasting, where we're simply sending viewing rays through each pixel and sampling the volume at discrete locations. Now, when we are looking at the data value profile that we will encounter along one viewing ray, then this may look something like this. Now, we think about what happens when we do MIP, so we are always taking the maximum along the array and showing it at a pixel, well, then the maximum will behave like this. So this is the only value that we actually display in the end. On the other hand, if we are doing direct volume rendering, the situation is a bit more complex. We have some sort of transfer function that specifies what is opaque and what is transparent. And based on this transfer function, we will accumulate certain opacity. And once we have accumulated full opacity, we cannot accumulate any more color contribution. So we cannot see through a completely opaque material in essence. So the accumulated color for a simple transfer function setting may behave in gray value like this. Now, what we see here is that these interesting events along the viewing rate, where the maximum changes are rarely or will typically not be captured unless we spend a lot of time tuning our transfer function. So what could we do in order to make DVR behave a bit more like MIP? Well, we can say, let's define a function that captures these events. Well, and a very simple function that does that is the maximum intensity difference. So we are simply taking the difference at the data value at the each sample along our viewing rate and the current maximum. So function will look like this for our example. And what we can now do is we can say goodbye to the physically given assumption that, well, nothing that is completely opaque or nothing can shine through something completely opaque, and say, well, what if the opacities are not monotonically growing? What if we reduce the opacity for something that is important, like these important events? So we want to reduce the accumulated opacity along the array in order to make it possible to accumulate more information behind those regions. And if we do that, then also the colors will change, and you can already see that the color profile I have here now very much looks like the maximum value profile that I showed in green. Now let's look at the outcome, and for this let's also be fair and use the same transfer function or the same color specification for DVR and MIP. So then DVR would look something like this, not very informative. And if we now use this new approach, this META approach, we will get something that really looks like a hybrid between the two. So we get all the context information, but we also get information about things that would normally be occluded. Now, what is the benefit of that? Let's look at an example. So this is actually a contrast MRI data set of the brain. And if you look at the MIP image, we see, well, there's a lot of blood vessels there. However, if we look at the META image, we also don't have to do any transfer function tuning. But if we look here, we actually see there's a small aneurysm that would really be difficult to detect in a MIP image. Here's just another example, another MRI data set where we now smoothly transition from DVR via META, our new technique, to MIP. And you can already see here in the center there is a rather large aneurysm, and using MIP it's much more difficult to judge the shape and extent of this aneurysm. Now, this was one example how we can incorporate a bit more information into our visualization algorithm by essentially including here data value semantics. We know that blood vessels will have higher data value, and we know that we're interested in blood vessels. So we gain some benefit, but still we only incorporated limited information. we do not really consider the geometrically arrangement of our focus object, the blood vessel. So the detailed assessment of the vessel lumen, the amount of blood that can flow through the vessel, is still difficult. So what else can we do? One concept that has been developed for this purpose are reformation views, such as curved planar reformation, already some time ago. And the idea here is that we essentially take a ruled surface and place it such that it intersects the center line of the blood vessel. And here's a short animation of how this looks like. This is our blood vessel. And now we can specify a ruled surface that we use to simply slice the volume. And it always is in the center of the blood vessel. Now, for the purpose of investigation of blood vessels, we will not display it like this, but we rather keep the surface always oriented towards the viewer and let the viewer rotate around the center line. So you always get a view of the vessel lumen. And this is quite an effective technique compared to, in this case, MIP, which makes it sometimes difficult to accurately judge pathologies. And what we are comparing here to also is DSA, which is an invasive technique using X-ray, where the patient is injected a contrast agent, and the difference between not contrast enhanced and contrast enhanced is investigated. And that's still sort of the gold standard, but of course it's quite invasive also. And we can see that we can recognize the same types of pathologies and can judge, for example, that whether there's a viable lumen. And we don't see that in the MIP image, really. However, we still have a couple of problems with this basic CPR technique. On the one hand, it's sort of pixel-based, meaning we're always sampling along scan lines, which means if we have a blood vessel as shown here on the right, the lumen will be displayed as narrowed, even though the vessel is not really narrowed. Hence, there's a restriction with respect to the vessel orientation and also with respect to the view direction. Vessel visibility here essentially means what happens if we have multiple blood vessels that are in front of each other in depth. Then we can't really deal with that in CPR. So for this reason, we can think about, can we specify what we want here from this visualization in a more fundamental form. So one approach is that we formulate our surface generation as an optimization problem. So what we want is that center line points closer to the viewer are considered more relevant, and that surface points closer to the center line are considered more relevant. So the first condition encodes that we do want some sort of visibility, and the second condition simply means that we are interested in the blood vessel. And then we can use a level of detail approach that to generate a smooth surface that also behaves well in the presence of small perturbation, for example, of our center line. And compared to CPR, we can then, of course, use, for example, a raycasting algorithm to visualize this surface, where we simply take the surface as a starting point. We have no problems with vessel orientation and direction, and also we get actually a sense of vessel visibility in terms of how their three-dimensional arrangement is. Now, let me show you an example. So this would be the CPR image, and here you can already see some of the issues with these discontinuities here. And here is a CSR image where you can, for example, see that here we also get information about which blood vessel is in front, which we can't really judge from the CPR in the presence of multiple blood vessels. And here is an example of how we can change the view direction arbitrarily. Now, this already is a further improvement, but we still have quite some problems here. If we really want to judge the lumen in such a reformation technique, we actually need to perform the full rotations, 360 degrees, so we can investigate every point inside the cylindrical cross-section. And that means, again, browsing through quite a few images, which can make it difficult if you have, for example, pathologies that only show up in a few of those images, and you need to mentally, again, put this back together. So one idea is, can't we produce a single image that captures the same information? And the approach is that we can think about, well, we are interested in information around the vessel lumen, so we could aggregate these rotations into a single image. One approach to do so that we developed is called Curvicircular feature aggregation, where the idea is that instead of looking at slices, we are actually sending around the center line multiple curved rays. And these curved rays are used to sample points in the vessel lumen. And here's just a comparison between 3D visualization, where we are shooting straight rays, curved planar reformation, where we are essentially performing a single cut through the vessel, but in along multiple angles typically, and CFA, where we are shooting curved rays. So how does this work? In brief, well, we are traversing the vessel along the center line, but not on along axis-aligned planes. And we're sampling in these concentric circles. And along each of these circles, we can now aggregate multiple properties. In principle, we could even do something like DVR or MEDA, but what makes a lot of sense in the context of our application is to determine the minimum and the maximum along each cross section along each curved ray. And now, how can you think about that this is displayed? So we are also straighten the blood vessel, so it's always in the center of the image. And the rows now are along the vessel, along the center line. And in the columns, we present the data values that we have aggregated along a progressively larger radii. And since the min and max operations are order independent, we can split the image in half and show the maximum on the left side and the minimum on the right side. And the resulting image will look like this, and I'll actually talk a bit more on one of the next slides why the minimum also makes sense. Will look like that, but of course within the blood vessel we get a very good view of the lumen, but the further we go away from the center line, the more difficult these images become to interpret. So what we also can do is we can split between a region that contains the blood vessel itself and a context region. So in the region in the blood vessel, we display the CFA as I discussed. On the outside, we can, for example, display regular volume rendering or MEDA. And in this way, we can also get anatomical context. Another thing that is possible is to also investigate the stability of the center line, since these are typically automatically determined, but of course these algorithms will have small variations, which can sometimes affect the visualization. So what we can also do when we are sending our curved curved rays. We can basically sample at multiple offsets from the actual vessel center line and aggregate and look at the variance for each curved ray. And we can also visually encode this. And that will essentially tell us something about how good is the center line, how centered is it in the blood vessel. This is shown here, where red means that there is a high variance. So these are areas where the center line is less than perfect. Now, how can we investigate whether this is actually effective? This phantom data set that specifically is meant to simulate quite a diverse set of pathologies. So we have a small off-center calcification, a concentric calcification, meaning that there is still lumen, but the calcification is around the vessel wall. A big eccentric calcification, eccentric soft plaque, which are also deposits, but they have lower data values, so it's not calcified, but they still decrease the vessel lumen. Then this would be a concentric stenosis. And then we have a small eccentric calcification where we try to simulate that the center line is actually off-center, that there was failure in the center line algorithm. Now, let's look at this data set using MIP. And here are multiple CPRs. So one thing that we can immediately see with MIP, for example, is that, well, the concentric calcification looks like a complete occlusion. We can't really assess this. And so does the eccentric calcification. So this is of course a big problem. In the CPR, we get a more detailed picture, but we also see that, well, depending on the angle, we sometimes don't see all of the pathologies. Well, here is the CFA image, which is also just one image, but it clearly lets us assess is there a viable vessel lumen, and shows us all of the pathologies. Now, just a quick view of how this looks in an actual application. So, of course, there's not just one blood vessel, but there is a whole vessel tree. And we can basically lay out this tree on screen according to the anatomy in a well-defined system, and also include what is called a transition margin. So when switching to one branch, from one branch to the next, that the certain region across the branching point is re-investigated just to make sure that nothing is missed. And then we can go along particular paths in this vessel tree until everything has been investigated. Now, here's an example where we have stenosis, which is visible here in the CFA view, and here we can confirm it using actual axial slice views. And in the CPR, for example, we see that it only shows up in 2 of the images, which makes it quite easy to be missed. And here is the CFA image, which clearly shows that here we have the narrowing. Just another quick example where we have an occlusion. And this is also clearly visible. I'll just give a quick view of how this looks in an application. So, of course, there are multiple linked views, and the CFA is used for primary navigation. And there is a linked slice view and also a 3D view for additional orientation. And we can do things like scale along the radius, so we get a more detailed picture, and of course change contrast and so on. Now, we have seen that we can already get some advantages by thinking about why has the data been generated, what are the structures of interest, and how do our visualization algorithms behave. But still, we end up with a set of techniques that maybe or may not be useful for certain types of examination. So we have different slice views, we have different Reformation views, we have different 3D views, and for a comprehensive diagnostic workstation, we still have to provide all of them. And that exactly defines how current medical workstations often look like. So you don't only have one monitor, you have typically 3 monitors at least, and in addition to the images, you have sidebars, you have context menus, and so on. So there are really many different views that the user can choose from, and worse than that, they are identified by weird acronyms such as CFA, MIP, MEDA, and so on, that a radiologist actually has no idea of what they mean, even if the words were written. These are terms defined by us, by technologists. And of course, there are many parameters, options, and settings galore that are modified in this side panel. But actually, this is a very unfamiliar construct for a radiologist. A light box where X-ray images are hung up does not have a menu or a toolbar. In fact, radiologists would really be interested in the images alone. So one approach is to make also the interface side closer to the users, to make the image the user interface itself and avoid all this additional scaffolding. And one way that we investigated to do that is what we call smart views. Essentially, the idea is that we already have a lot of information in our input. We do have the CTA information. We do have, this is generated on a routine basis, semi-automatically, a bone mask, a vessel mask, and a vessel tree, just to do things like CPR. And then we add a component that allows us to say, well, different types of tissues are best visualized using different techniques. We can, together with the domain experts, set up such a rule base. And what we could do is we could use this information to essentially decide, well, which view is most appropriate for which part of the image. And then we could automatically suggest these views to the user. Now, the question is, how can this work? And I'll quickly go through one option of how to do this. Essentially, our input data, as I already mentioned, contains this information about different types of tissues and about the vessel tree itself. And our user interaction should be very simple. So the idea is that the user only has to specify using his mouse cursor what he's interested in, and from that we want to deduce what is the best way to show this part of the image. So we have a circular region of interest, simply defined by the mouse cursor location, and within this region of interest, we take a look at every pixel and determine, well, what is the corresponding tissue for this pixel, and we additionally also weigh it with the distance from the center. And this this is all the information that we need from the user side. And then we have a set of rules that define the relations between the input and the output. So if we have a blood vessel, for example, we would like to show a Reformation view. If we have tissue. So the question is now, how do we get from these multiple input layers that we have from our region of interest and the output views to these output views? So we have a rule base, and this rule base should be ideally as easy as possible so that we can design this together with the domain experts. So our choice was to use a simple human readable rule base where we actually specify this in rather easily understandable if-then rules, and then evaluate these sort of rules using a fuzzy inference system. So basically, these vessel layers that we define in high are fuzzy set membership functions. And the fuzzy inference system will then give us essentially a ranking of how good is a view for a particular region of interest. And they'll not detail how this works, but we use a standard fuzzy inference system. Just a simple example. So depending on whether we click on a blood vessel, for example, where we get oblique slice, the correct oblique slice that shows a good view of the vessel, and a Reformation view, or we are more on the tissue side where we get volume rendering as suggested. And these are interactive views that the user can interact with also these integrated views. Now, these sort of integrated views are very useful, and that's why I want to show as a last example another application of such integrated views, and this is in the context of segmentation, because often we need to segment structures, but fully automatic segmentation is quite difficult in biomedical data. So here we can also incorporate structural information to allow the user to easier correct segmentation errors, which is usually done simply by manually correcting contours in the segmented images. And the information that we use here is actually information provided by an automatic algorithm. We base this on the assumption that the automatic algorithm will at least provide us a good approximation of the shape of the object to segment, in this case, for example, a liver, and we can then take a look at the structure and detect likely candidates for wrongly selected regions, and then provide integrated views for the user to correct the segmentation. And the idea is that we can the structural information that we use is essentially a skeleton of the object, where we collect regional histograms and evaluate their dissimilarity in order to provide correction scenarios. And am I still there? The power was already was again fluctuating. So here's a quick demo of how this works in practice. So we here see incorrectly segmented region, and we get these automatic suggestions for regions that are probably not correct. And by simply clicking on the region, we get a preview of what will be removed. And it is where we can very efficiently correct the segmentation. Now, let me conclude this talk by first summarizing again what I mean by smart visualization. It's essentially using the right tool, the right algorithm for the right job. And key is here that we know what the right job is, what is the task of the user, but that we also, and this is sometimes not considered enough, what the behavior of our algorithm is. And the challenge is to, of course, acquire this information in a more systematic manner. I showed examples of how this can be done in for specific applications, but I think for the future, there are a couple of things that are really big challenges, but also great opportunities in making visualization more smart. For one, ideally, a smart system should become smarter over time. So there should be some form of learning. And one way to do this is to simply incorporate mechanisms to capture data, whether something was successful or not. Did the user switch the view again after the view was suggested or not? And then we can employ recent significant advances in machine learning, such as deep learning, to attempt to improve our system based on this user interaction. And we could also use this acquired information for a more goal-directed exploration of the algorithm space. What do I mean by that? Well, if we see that a particular type of algorithm is always used in combination with another type of visualization algorithm, then it may be worthwhile to think about developing sort of a hybrid method, as I showed you with the DVI MIP case. So we can also use such information as a goal for what algorithms still need to be designed. And with that, I would like to thank you for your attention, despite some technical difficulties. I hope at least some of you are still there. I see the people in Linz and maybe online too. And I, of course, want to thank my collaborators for the work that I've presented here, and I'm happy to answer your questions.
Okay, thank you, Stefan, for this very interesting, I should say smart talk, probably.
Except for the electricity.
Yes, is that a regular problem in Norway?
No, but apparently we would need a smart grid, I guess.
Yes, I guess so. Okay, although we are already a little bit over time, we have some more time for one or two questions. Any questions to Stefan? Okay, I should probably start. So you said maybe it's possible to learn these rules and to learn the tasks. How can you, as a system, know that the task was successful or not? Which is very important for learning. Yeah, I mean, you said you could wait if the user has switched the window, but maybe you need some kind of annotation mechanism where the user can tell the system that this was the right choice or a bad choice.
Yeah, I mean, that's a very good question. I mean, there are multiple ways that one can think about, but in the most simple case, this can be fairly unobtrusive. Facebook is able, just from a very simple like button, by collecting a lot of data, have quite detailed information about the preferences of people. And similarly, just something as simple as a like button could already help to get a bit more information. Maybe it's even possible to do this in a more or in a finer grained manner. But I think for most applications, it needs to be something unobtrusive. So of course, it can also be something like, you know, what is the frequency that a technique is used in conjunction with other techniques? You know, how long is a view used? For instance, if a user switches through different views and one is only shown for a really short time, probably it's not relevant.
Yeah, Are you
of course, there are many challenges in designing interfaces that are not obtrusive but still capture sufficient information in order to really get something useful out of this.
Yeah, are you already working on this learning?
We are looking at some aspects of this, but of course it's also not trivial. One of the challenges also that things like Facebook also do not have to get sufficient data. Visualization is often only used by a comparably small number of users. So you don't have the power of numbers yet, but since more and more things are also more and more visualization applications are also put online, there is an interesting opportunity there to also get more people and more data from other places that otherwise you simply would not have access to.
Yeah. Okay. Well, thank you. Second question. Last question. Okay, I have a bunch of questions, but I will only pick one. We can discuss the rest next week in Chicago. Is the techniques that you discussed, the CFA and the CPR, are these invariant to the size of the structures? So let's assume you have very tiny vessels. Let's assume the resolution of the data set is big enough, and you have really large vessels. Do you need to tweak the parameters to make them work for the different size of the structures?
I mean, it's an interesting question. I mean, usually these sort of techniques, they are not really used, or peripheral vessels are not so interesting in this context. You're usually using this for major vessels, and for peripheral vessels, other techniques that are specifically designed to capture perfusion information are used. But in principle, within at least the standard range, they are relatively invariant. But they are relatively invariant and work independent of the vessel size.
Okay. Well, thanks again for the great talk. A quick announcement: the next talk will be on the 10th of November. Piotr Didik from Saarland University and also Max Planck University will be talking about new output devices. I'm curious what that will be about. Yeah, for those of you who did the seminar, which is you, who else? You, and also the third person, and you. Okay. Yeah, thanks for coming today, and also thanks for coming to all the rest. Stefan, have a nice evening, and I hope without any further power outages.
Well, now look who's playing with the generator.
Okay, great.
All right.
Yep. Bye. Thank you. Bye.
Bye.
Could you stitch it to one video then and put it online?